You guys have a PRR assignment that is due today. Due today by 5, so a bunch of you have emailed it, which is exactly right. Yes, there is no thing on Moodle, just email it to me. I'm not going to talk about it today because it's not due till 5 and everyone hasn't turned it in yet, so that would be kind of rude if I started talking about it now. So I just wanted to mention all that and give you the reminder on that front. In addition, if you have been looking for last week's lectures on YouTube, they are not there yet. Super exciting, more technical issues. In fact, my microphone apparently was actually completely broken that I was using. So what I'm going to have to do is so you're not going to get, the ones on YouTube are not going to have like our excellent class discussions and jokes. I am basically going to have to go home and re-recite them to my cat. So the recordings, she knows so much immunology at this point, it's amazing. So I will get that done and get those posted. I also have a new microphone, so hopefully no more issues. So the plan for today is to finish talking about a few aspects of the innate immune response, specifically something called the antiviral state, and then transition into thinking about antigens and antibodies. We have been talking about this whole process of inflammation over the past few classes, where of course we have our local macrophage that is detecting a microbe, and in this case it is a bacteria, using some kind of PRR, making classic inflammatory cytokines like IL-1 beta, IL-6, and TNF-alpha that make sort of the general inflammation changes that we've seen, recruit in neutrophils, and those neutrophils will phagocytose that bacteria and get rid of it. As we discussed last time, sometimes we need a different response when the microbe is a virus as opposed to a bacterial pathogen. One reason is because one key thing that viruses have to do is actually figure out a way to get into cells, and sometimes phagocytosis is actually doing their job for them. So phagocytosis is not always the best thing. In addition, oftentimes the problem in a viral infection is the infected cell, and so we're not going to have a neutrophil or some other phagocytose one of our other cells that's infected just on a volume issue. There's also not a whole lot we can really do when our cell is infected, that we can't sort of cure that cell, get rid of that infection in that cell as once the infection gets particularly far along. Someone asked me last time, what about apoptosis? And the answer is, yeah, there are actually a bunch of ways that cell may end up going to apoptosis. Both direct and indirect ways, which we can think about at some point. But there's nothing we're going to do to cure that cell. In fact, this cell needs to die, perhaps by apoptosis in some way or another. But more importantly, we also want to prevent something bad from happening. So the idea is sort of that this cell is potentially going to get killed, but really what ends up being important is this cell wants to tell its neighbors, hey guys, there's a virus around, get ready, to make sure that they can block viral infection and that they don't get this same fate. In 1957, some investigators named Isaacs and Lindemann were looking at cells in a dish that were infected with viruses. And when they took these cells in a dish and infected them with virus, they saw that the cells made a new product and put that new product into the media. So they secreted some product. And when they took that secreted product and put it on other cells, the other cells couldn't get infected anymore. So somehow the infected cell made a magical product that blocked other cells from getting infected. Except they didn't usually say blocked, they said interfered. It interfered with other cells being infected. So they called this thing that they didn't know anything about interferon, because it interfered with other cells' ability to be infected. We now know that there are 3 big types of interferon: type 1, type 2, and type 3. When we're talking about innate immune responses, we are talking about type 1 interferons, which are officially interferon alpha and interferon beta. One could, when it's talking about innate immune responses, also talk about type 3 interferon, which is interferon lambda, although I'm really not going to say much about it. I'm going to be focusing on alpha and beta. Type 2 interferon, which is interferon gamma, is an adaptive immune system thing. And we're going to deal with it when we think about the adaptive immune system. So really, right now, we're thinking about type 1 interferon alpha and beta. You can see these are sort of these general interferons, alpha, beta. Both of them are type 1. I often will put them together as just interferon alpha, beta. You do not need to worry about distinguishing between them. And they are, you can see, induced by viral infection. They are made extremely rapidly following infection by a virus-infected cell. And in fact, in many cases, they can start to decline by 10 hours. They are going to be made over the course of hours. We're not usually going to see them on the course of days. In my research lab, we do a bunch of work trying to stimulate interferons and measure them. And in general, they're gone by 48 hours in our hands. And so we can see kind of this type 1 interferon process in this figure from your textbook. I've got a couple of other figures that are going to get us towards similar things. And so here we can see this cell that is in yellow. It's our virus-infected cell. We can see the virus coming in and infecting the cell. And it's not shown here in great detail, but that virus can be recognized by a PRR. There's a PRR recognizing some part of that virus. One of the types of signaling will be a change in transcription, just like you saw with other PRRs that we saw looking at bacterial responses. But here, instead of making the inflammatory cytokines, IL-6, IL-1 beta, and TNF alpha, we're going to see the type 1 interferons, like interferon beta or interferon alpha being made. And so now you can see this cell starts making interferon. And that interferon, I'm going to just look over here at this one on the right. This one in the middle is true, but I'm going to ignore it for the moment. That interferon beta will float around and go to a neighboring cell and will bind to an interferon receptor in that neighboring cell and turn on a response in that cell. And that cell is now going to, that cell is uninfected. There's no virus in that cell, but that cell is going to change its biology so that it now is going to go into something called the antiviral state, where it can block viral replication. So the virus is not going to be able to reproduce in that cell because that cell is in the antiviral state. And you can see another version of this from another textbook here. Here we've got our infected cell. The virus is in it. It's making new viruses. It's a virus production factory. It's a goner. There's nothing we can do for this cell. But it's going to make some type 1 interferon. That type 1 interferon is going to bind to the type 1 interferon receptor on a neighboring uninfected cell. And this cell is going to be in the antiviral state. So as you can see, if the virus tries to enter this cell, the virus is going to be blocked. We're going to block replication of this virus. And so you can see this sort of overall process that's going on here. when we think about what's going on here, you really want to know kind of what in the heck am I talking about is this antiviral state, this magical antiviral state. It's something that happens in the cell as a result of interferon binding to the interferon receptor. And the big picture answer is that this uninfected cell starts doing a lot of transcription and starts transcribing a lot of genes. There are thousands of possible genes that could be transcribed. We think that the different patterns and exactly, in this case it's number 4, number 57, and number 73, versus in some other case it's another pair, is important, but we don't totally understand what's going on there. But we do know that all of these things that are made are called ISGs, the interferon stimulated genes. So when interferon binds to the interferon receptor, that cell now begins to make interferon stimulated genes. And the combination of which ISGs are made in any particular situation is probably important, although it's not super well understood. One thing that we do know is that most of these interferon stimulated genes, ISGs, have in common a particular promoter. They all can get turned on by the action of a particular promoter, which is called the ISRE, or the interferon stimulated response element. You can see we're very creative with all the naming. And so that's sort of the big picture of what's going on with the antiviral state. But as you might wonder, what the heck is the mechanism? How does the cell suddenly have the ability to block viral infections? Well, one answer is that it depends on which ISG we're talking about. And there's like thousands of them. I can't tell you what all thousands of them do. Even if we knew, we wouldn't have enough time to talk about what all thousands of them do. But I'm going to tell you about a couple of highlights. I've got a few on the next page that are sort of really key important ones. I might verbally mention a couple other ones just to kind of give you a big picture understanding of what's going on with this antiviral state. So here you can see interferon, type 1 interferon binding to the interferon receptor. We're going to get transcription of interferon stimulated genes, and 4 of them are shown here: protein kinase R, 2', 5' oligoasynthase. We can see MX, we can see IFIT. I'm mostly going to talk about these 2, protein kinase R and 2', 5' OAS, as my examples. So when we make this ISG, this protein is able to eventually lead to an inhibition of translation. And so basically when this protein is activated, some changes happen in the cell that inhibit translation. And that's awesome because the virus needs to make new viral proteins if it wants to be if the cell is going to be a virus production factory. So if we inhibit translation, the virus can't make any new viral proteins, and we foiled it in its tracks. Insert something about, Scooby-Doo. We also have this molecule called 2', 5' oligoasynthase, which eventually leads to mRNA degradation. So again, pretty useful. The virus needs to have mRNA to get translated. And so if we turn on the production of an enzyme, this RNase L that's shown here, that actually chews up RNAs, then the virus isn't going to be able to reproduce very well. There is one that is involved that's not shown here that is pretty cool that messes with protein stability. There's one that is really cool that messes with how many dNTPs there are in the cell, so you can't make new DNA or RNA because you got rid of all the dNTPs. So you can see that it's sort of a multi-pronged attack on the virus. And if we had sort of gone through lots of details of interesting virology and how some things of viruses work, I could tell you all sorts of cool ISGs and some of the cool pieces that they do. But here you can kind of see big picture. But yes, Jazz.
This may be a silly question, but the protein kinase R, the 2', oligoasynthase, those are all examples of ISGs.
Yeah. Yep. Those are examples of ISGs, exactly. So sometimes when people hear about this process, they hear about the antiviral state, they really don't like it. They really don't like what we're seeing here on this slide. And they say, so here's the problem. I would like my cells to be in the antiviral state all the time. Why were they ever able to get infected in the first place if antiviral state is a choice? Why are we ever not in the antiviral state? Becomes this question that people will ask. Yep, David.
A lot of the things that happen in the antiviral state is also prevents stuff that you need to survive. For example, DNA synthesis. You prevent mRNA production. You prevent protein, I think, changes.
Mm-hmm.
So all of those use your cell needs.
Exactly. And so David has pointed out correctly, this says inhibition of translation. It doesn't say we inhibit viral translation. It says we inhibit translation. We stop all translation in the cell. So that means the cell can't do any of the translation it needs for its normal cellular processes. This says inhibit, our mRNA, or this is mRNA degradation. It's not degrade viral mRNAs, it's degrade all the mRNAs. So again, we're going to be messing up the cell and really making the cell unable to do a lot of its normal processes. And so if you think about this, that's not really ideal for the cells. So what do you think sort of is going on in sort of a general biology of a cell in the antiviral state? Is it a very healthy cell? No, that cell is undergoing the antiviral state is not healthy. It's not able to do a lot of normal cellular processes, but it's going to live. And so the idea is if you have your cells in a really brief antiviral state for maybe some hours, you can, while the virus is around, you can stop virus replication. But then you need to have that cell go back to normal, go back to its normal processes so that it can do normal cell stuff. So one of the things that you can see here is that basically all cells can make type 1 interferon. So this says most, if not all. We'll just say all. All your cells make type 1 interferon. Basically, all of your cells can say, hey friend, there's a virus around, get yourself ready. Which of your cells do you think should be able to listen for that call? Or alternatively, in biochemical terms, have the receptor for interferon? Who should be listening to have this? Who has the receptor? What do you think, Emily? Everyone. So all of your cells have receptors, just like all of your cells can make interferon. So interferon can induce some genes that are not great for cells. Basically, all of our cells have receptors. And so if we start making interferon in our body, we generally feel it. We generally know about it because we are basically putting some of our cells in a pretty bad biological situation. Generally, every viral infection makes interferon. And when that interferon is made, you start having things like fever, chills, nausea, and malaise. Sometimes people talk about those as flu-like symptoms. They're not flu-like symptoms. They're I'm making interferon today symptoms. Sometimes referred to as acute viral infection symptoms. I got vaccinated on Friday. I was a great poster child for some of these, particularly on Saturday. And so sometimes I think about interferon. One of my other textbooks shows the idea of interferon as sort of, there's a forest fire, And the way that you deal with the forest fire is that you cut down a little bit of forest. Nobody likes cutting down forest. Nobody wants to cut down forest. But if you cut down some forest, then the fire can't keep spreading, and the rest of the forest you didn't cut is safe. Like if you just cut down a little stripe of forest. That's what interferon is. It's not good for you. Just like cutting down part of the forest isn't good for you. But it's sort of making an area of cells that can't be infected to stop the virus from sweeping through the body. You feel crappy. I am well aware because I did this weekend. You can also, there's a scene in Dallas Buyers Club where one of the characters thinks he's going to treat his HIV with interferon injection. And so he actually gets interferon in another country and injects himself in a bathroom stall in an airport and then has a heart attack. So there are some situations where, yes, interferon is now used clinically, but in general, if you got a lot of interferon in your body, especially too much or unregulated, too much, too long, we're not doing Goldilocks, it is bad, 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 bad. And we also know that there are a lot of interesting sort of interrelationships between some of the different immune responses that are out there. So if I was going to be very, very oversimplified, in the top, we're really thinking about our pathogen as being bacterial, while in the bottom, we're really thinking about a viral pathogen. In the top, we're making the sort of classic inflammatory cytokines, TNF-alpha, IL-1 beta, and IL-6. In the bottom, we're making the classic antiviral cytokines, interferon alpha and interferon beta. And in the top, our results are recruiting phagocytic cells and improving phagocytosis. In the bottom, our results are the antiviral state. Now, I am showing these two things here as a very strong either or. They're not an either or. They actually both happen at the same time. I probably had some IL-6 and TNF going on this weekend too. But for your purposes, you can think of them right now as being sort of one or the other. In fact, part of the problem in COVID is actually that the balance between the two is messed up, but because both of them really are going on. But that's okay. We're not going to go all the way to that right now. And one of the other things that is relevant here is that clearly what you can see from this distinction is that the cell knows what kind of microbe it is. The cell knows, is it bacterial, is it viral, and starts to push the immune system down a particular path. That ends up going back to which pattern recognition receptor got stimulated. And so it goes all up, it's which PRR is stimulated. That kind of helps the cell decide which type of response should I initiate. Should I initiate this more inflammatory response, this more viral response? And it pushes the whole immune system sort of down a direction of what kind of response would be the most useful. And in fact, that even relates to the fact that, I didn't show you a whole lot of detail of PRR signaling, and I'm not going to, and it's breaking my heart as a person who studies PRR signaling. But in fact, basically different PRRs give us different transcription factors. One transcription factor, NF-kappa B, gives us those inflammatory cytokines and gives us all sorts of things that are really useful for inflammation, while a different transcription factor gives us interferons in the antiviral state. So it's all about which PRR do you have, what kind of signal comes through that PRR to what kind of transcription factor, and as a result, what kind of product is made. Also, as you can see listed here, it's not listed on the interferon side, but it's also true. All of these innate cytokines are going to really set up the adaptive immune response and the quality and the type of the adaptive immune response. And so this is a bit about what you're looking at in the homework for today. What we've realized is that it seems like you need sort of some information about the microbe and some information from the PRR together in order to get a good response. And if you get those individually, it doesn't quite work. One way that some people have talked about this hypothesis is some people say you need something, your immune system doesn't just have to see a protein that might be foreign. It needs a protein that's foreign plus a signal that says danger, sometimes known as the danger signal, to say, here is a protein I found, and also there's something wrong with it. It has stimulated a PRR. It tripped a PRR, and I have this protein. So there's clearly something dangerous because it tripped the PRR. So we're going to give a danger signal and a foreign signal. And that's what's going to induce the immune response. And so you can see it's sort of that same idea of combining these two signals. That's something that we're going to, of course, talk about a lot more as we move forward. Later in the semester at certain parts of adaptive immunity, and it's also very relevant to the stuff that you're reading about for the homework for today. But speaking of adaptive immunity, at the top we can see the immediate innate immune response. That was our innate immune proteins that could act in the first 4 hours or so. Maybe they win, maybe the pathogen wins. The pathogen's still around, then maybe we need these cells to attack the pathogen, and maybe that's good enough, and we're done. Or maybe the pathogen is not eliminated. And now it's time for us to have some adaptive immune responses that start acting against the pathogen. One thing to note is that the adaptive immune response was doing a bunch of stuff before it actually dealt with the pathogen. So there's a little bit of a delay, and there are actually actions that are having to happen with the adaptive immune system before we're going to be able to get into our pathogen. But I want to start our discussion of the adaptive immune system with some classic experiments where people were trying to understand some things. They didn't know it was called the adaptive immune system then, but it helps us segue into where we're going. Which are a set of experiments done by scientists named Van Barry and Kitasato. The experiment, I'm going to tell you the story as if there's like one paper, but actually there's a whole series of papers that I'm kind of mushing together right now. Van Barry and Kitasato won the Nobel Prize for this work. In fact, you will see throughout this semester so many Nobel Prizes for so many experiments that I'm going to tell you about. And one thing that all of those experiments have in common, and Van Barry and Kitasato is no exception, is that in some cases we now know they did something really lucky. And if they had done the experiment in a slightly different way, it wouldn't have worked and would have been a little bit of a mess. And so the reason why I'm telling you that is the thing that Van Barry and Kitasato did that was really lucky is the particular microbes they used. So they were trying to describe the immune system. And they picked 3 particular microbes. It turns out those 3 microbes all are dealt with by the immune system in the exact same, very simplified way. And so they got a kind of simple answer. So just know, I'm going to tell you the Van Barry and Kitasato experiment and go through the whole thing. There are other microbes where you could do this experiment and it wouldn't be so clean. So don't think that like this explains everything, but it does explain a lot for these 3 microbes. So the 3 microbes that they used were Clostridium botulinum, which is the causative agent of botulism. Clostridium tetani. That is not how you spell that. Maybe. I don't know how to spell it. Doesn't matter. Whatever. It's the causative agent of tetanus. The other organism that they used is Corynebacterium diphtheriae, which is the causative agent of diphtheria. I'm honestly really only going to talk about these two in my example today. But if you actually Google this, for some reason, Google really likes to talk about the diphtheria side. Whatever. So von Behring and Kitasato first did an experiment, and they did this experiment with rabbits. And one of the other great parts about talking about this experiment is you get your first taste this semester of my awesome artwork, because I am such an artist. So they did an experiment with rabbits. And they had two different groups of rabbits to start with. One rabbit that's how you spell that. Why? That's right. I don't know what was wrong with me over there. They did them with these different microbes, and they did them both at a low dose. And when they did these experiments, the rabbits lived. We know they lived because they could eat some carrots. They also did basically the same experiment with these two organisms at a high dose. And when they did those experiments, the rabbits died. So low dose, they live in happily ever after, get to eat carrots. High dose, they died. So, okay, great, fine. Now, they only have rabbits left over who already got a low dose of these bacteria. I shouldn't totally erase this, but we'll figure it out. So they took these rabbits, and they gave them both, again, I'm oversimplifying like many years of experimentation here, a second infection. And in this case, we'll say here that it is C. tetani high. So a high dose of tetani to these rabbits that had already received low dose, either of C. botulinum or C. tetani. And when they did this experiment, this rabbit lived and ate carrots. And when we did this experiment, the rabbit died. And so the first thing they figured out is that there was this ability to get some immune protection from a previous infection. And that that protection was specific. So it had to be a previous infection with the same microbe. You can see they used a different microbe, and in fact they're still pretty closely related microbes. They're still in the same genus of Clostridium, and that protection didn't work. So this was sort of the beginning of what immunologists think of as this property of specificity. So the adaptive immune response could be specific in its protection. So that was pretty cool. It wasn't just protecting you against everything. It wasn't like rising tides brought up all the boats. It was only a very specific improvement in immune responses. So they're pretty excited by this. But then they went on to do an additional experiment. And this is the part that would have gone differently if they used different microbes. So they took a rabbit who had gotten a low dose of Clostridium tetani. And the hot debate in the late 1800s in thinking about the immune system is the immune system made of cells or was it made of liquid? I told you about cellular versus humoral immunity. They were sort of like, is it in the blood or is it in the cells? And so they took this rabbit and they took 2 different samples from it. One sample was the spleen. And basically what I want you to think about with the spleen is that the spleen was cells. It was a big bunch of cells. So really, when we think about spleen here, we think about cells. Or they took blood. But they didn't take just the whole actual blood together. They took a part of the blood called the serum. And that's what this slide shows. So you can actually separate blood in a bunch of different ways. One of them is that you can let the blood clot, and you'll get basically the cells and the clot at the bottom, and all of the liquid, which contains all of the proteins and other stuff, at the top in the serum. So they took either the serum, which is basically the liquid part of the blood, or cells from the spleen. And they put those into new rabbits. These rabbits had never before been infected with any of these types of Clostridium. So basically, these rabbits were either getting experienced serum or experienced cells. And the question they were asking was, is it the cells or is it the serum that actually protects you, that provides protection in this case? Again, if they did not pick these particular microbes, they would have gotten a messy answer here. So I'm going to tell you the answer, but please do know this answer is for these particular microbes, but it is how we figured stuff out. The rabbits that got the serum lived, ate a carrot. The rabbits that got the cells died. And so Vomberi and Kitasato said there is some magical, awesome thing in the liquid part of the blood, in the serum, that leads to immune protection. And we can transfer it from one previously immunized or previously protected rabbit to another. We can actually take serum from an experienced rabbit, give it to another rabbit, and that rabbit will be protected. Yep, David.
So the process of giving the cells in the spleen to a healthy rabbit killed it? Or was that healthy rabbit?
No. Oh, I'm sorry if I wasn't clear on that. They also did an infection.
Okay.
They did high dose Clostridium tetani. Sorry, I didn't mention that. So here we had protection from the infection. Here we had death from the infection. I apologize for not making that clear. And so the idea was there's something magical in this serum that was protective. You might wonder, I guess spoiler alert, is that I'm going to tell you that this magical thing in the serum that came from these experiments was the antibody, was understanding antibodies. And the reason why I'm pointing out that spoiler alert right now is that you might look at this and be like, oh my God, why is she telling us 1800 science? No one cares. But in reality, we still treat some people with antibody transfers, just like you saw with these rabbits. This is known as passive antibody therapy. So we can take antibodies from someone's blood and put them into an individual and protect that person. This is, like I said, is known as passive antibody therapy when we do this in humans. And so if you were to have a black widow spider bite or rabies infection or a snake bite or a few other things, tetanus, as you can see here, you would actually be treated with antibodies made from someone else. This is still done to this day. during sort of peak COVID in 2020, the first thing that we did to treat people was we took blood from COVID survivors and did passive antibody therapy. So we had people who survived COVID, and they were like having drives in New York all the time to have people who lived through COVID give blood. And they were basically taking the antibodies and putting them into patients to try to help those patients. So passive antibody therapy is very much still something that is used now. And in fact, after this was going on for a while, pharma companies figured out a way to just make the antibodies and not have to get them from a person who had been infected. And so we started using monoclonal antibody therapy, which was just passive antibody therapy where we made the antibody in a lab. And so there were a number of different antibody drugs that people would get to help them when they had COVID. It was really the same exact process of passive antibody therapy that goes back to von Behring and Kitasato, just a bit more technology in terms of how we made the antibodies. But if we go back in to thinking about these experiments, we can look at the von Behring and Kitasato experiment, and we can see, okay, so the first thing they did is they found out the magic stuff was in the serum of the blood. But that's all they knew. And so the next thing that they had to do was they wanted to figure out, and now when I say they, I say past scientists, because I don't think von Behring and Kitasato did this experiment, but definitely the next one they didn't do. I can't remember who was doing the experiment at this point. The idea was they wanted to know, is this magic thing a protein? Is this magic thing a lipid? Is this magic thing a carbohydrate? Is this magic thing a nucleic acid? So how might you figure out the answer to that? Yep, David.
So you have that serum you inject, you put something that digests one or two specific types of biomolecules in it so that everything degrades except for that one lizard.
Yeah, so really what they did is they did this experiment, again, as David pointed out, but first they added a protease, and that got rid of all the proteins. And then they're like, does it still work if you get rid of the proteins? Does it still work if you add a lipase and get rid of the lipids? Does it still work if you add an enzyme to digest all the carbohydrates or to digest all the nucleic acids? And so they did those types of things, and they came up pretty quickly with protein. That the magic stuff was protein. Like, okay, magic protein in the blood. Got it. And then someone was like, all right, so now what we want to do is we want to find out what the protein is. We want to like further break things down and find out what the protein is. And so they did an experiment that kind of looks like this. You guys in Bio 250 and those of you who've taken biochem have done SDS page gels, where you separate proteins based on size and you see bands on the gel. They hadn't invented that yet. And so what they actually did was doing like chromatography in like fractions. And so they found that there were 4 places where there were important fractions of the proteins because they didn't have a gel yet. So in some ways you can imagine these as like 4 different bands on the gel. And they found that when they did the separation, blood had 4 protein types in it. Now we know that they're just groups, but like whatever. One of them, there was just a ton of in the blood, and it was called albumin. Great. There's a lot of albumin in the blood. We're not going to care about albumin. And the other 3 kinds of proteins were globular proteins. That means they were secreted. Again, no one cares. I'm telling you this for exactly one reason, that they are globular proteins. I'll even write the word down. And there were 3 globular protein types. And they didn't know what they were. They didn't have any clue what was going on. So they called them alpha, beta, and gamma, the first 3 letters of the Greek alphabet. You will learn this semester, immunologists, oh, we love us some Greek letters. I did not know that being in a sorority in college was going to help me be an immunologist because of my how well I know my Greek alphabet. So they called one alpha globulin, one beta globulin, one gamma globulin. So alpha globulin, because it was the alpha globular protein. Beta globulin, and gamma globulin. And when you look at this plot, ignore the black line. The black line is an interesting experiment, but I'm not going to deal with the black line right now. They just basically, the point I want you to see is they got 4 different types of cells, of proteins. And then they repeated this experiment again. They said, is albumin the protective one? Is alpha globulin the protective one? Is beta globulin? Is gamma globulin? Which one has the protective effect? And they found that all of the protective effect was in the gamma globulin fraction. If you go to a hospital now and get certain types of treatment, you might get gamma globulin infusion or gamma globulin treatment. That's still what it's called at the hospital right now. Gamma globulin is just getting antibodies. That's all that means you're getting. You're getting passive antibodies if you're getting gamma globulin treatment. And so this is where that idea all came from. Gamma globulin was, after this, known as the immune globulin, the one that had the immune powers. People also shortened that to immunoglobulin or IG. And that is the name people tend to use for antibodies. So a technical term most immunologists say when they mean antibodies is immunoglobulin, or IGs. And again, that comes straight from this information from way back in the day. So also pretty early on, people wanted to figure out what did the protein look like? What was the sort of structure of this immune globulin or this gamma globulin protein? And they came up with some pretty cool experiments. So these are actual images where they could find this protein, and they always saw that it was shaped like a Y. So you can see the Y shape very clearly under an electron microscope. And they could see that there were sort of 2 identical arms of the Y and one kind of tail part. And they could also see that when they looked at those 2 identical arms, those were the ones that bound to stuff. Like those were the ones that bound to whatever thing the antibody attacked. And they were identical, while the tail was not so much part of it. And they did more biochemistry and got some more information about the antibody structure. And this is sort of the big picture of what we now know in terms of antibody structure. We're going to say much more about this today and also on Wednesday. So you don't have to be all on top of this right now, but know you've got a Y-shaped structure. You can see it's made of a few different protein chains. And you can see that we've got these 2 identical arms that are going to be binding something, as well as this tail region. Before I can go further into details of the antibody structure, I need to take a little side route. And the side route is that I need to tell you about the thing that the antibody binds to. Because I'm saying this is the part that binds the thing, but I haven't said anything about the thing yet. So we're going to take a little side route, talk about the thing, and then we'll come back to the antibody structure. And so the technical term for the thing that's bound to the antigen or the antibody is called an antigen. Somebody once upon a time, a long, long time ago, was like, look at this, it generates an antibody. So gen, anti. So they made antigen because it would generate an antibody. And you can see an antigen binding to an antibody here. In reality, the term antigen means that something that's recognized in an adaptive immune response. So remember, an adaptive immune response has specificity, and we are going to see response to a specific antigen. And so when we get to other adaptive immune cells and other adaptive immune molecules, antigen is still going to be the word we use for the target. So you could compare and contrast that to MAMP, which was the target in the innate immune system. Antigen is the target in the adaptive immune system. But if you look at this, well, it's shown on this image, but we're going to also see this image again. And then there's another really key sort of experiment that is relevant to thinking about what we know about antigens and antibodies. And this was done by a scientist named Carl Landsteiner in 1900. He wrote a book called The Specificity of Serologic Reactions. I have not read it because I did find a copy in a library once and it was in German. And that kind of ended that ability. But I can still tell you approximately what Landsteiner talked about. So Landsteiner was a chemist, and he was making different types of chemical molecules. And so what I want you to think about in terms of Landsteiner is he's making these new dyes that basically had never existed before in nature, or at least assume for our purposes right now, these different chemical compounds had not existed before. He actually was making these dyes and they were bound onto a protein, which in the end actually becomes kind of important, but we don't care about also right now. So he's got this dye attached to a protein, and he's got these 4 different dyes that he makes. So you can see he makes an aniline dye, which is shown here. He makes O-aminobenzoic acid, M-aminobenzoic acid, P-aminobenzoic acid. Their structures are shown here. You don't need to know all the details of the chemistry to think about that. What I want to ask you is, if you look at the pictures, what's the difference between these? Yeah, Brooke? Oh, sorry. What's in different positions on the structure? Yeah, so we've got this carboxyl group in different places relative to the amine group. Okay? And remember, this is attached to a big, ginormous protein. So that's actually like a pretty small difference in the molecule of just like, that angle of attachment. And when Landsteiner injected all of these into rabbits. And then he actually looked at the serum of the rabbits. And he found that the rabbits who got injected with aminobenzene made antibodies against aminobenzene that didn't respond to these other ones, even though they only have a pretty teensy little difference. Same thing, the aminobenzoic acid exposed rabbits only made antibodies to aminobenzoic acid that could distinguish. And so basically what we get from Landsteiner is, and also, like I said, at least what we're going to assume at the moment, is these dyes had never been synthesized before in all of the history of life. So it wasn't like these rabbits had been infected with the dyes before. It wasn't like some rabbit precursor in evolution evolved that it had to respond to dye. And what we learned from Landsteiner is, basically, you can make antibodies to literally anything. If there is a compound that goes into your body, you make antibodies against it. And you evolved to have that extraordinary breadth. But also this extraordinary specificity, where you can have antibodies that can distinguish these very small chemical differences. And so the idea with Landsteiner is we're seeing just how many different antibodies an individual can make. In different places, we quote different numbers. I'm just picking a number. We don't actually care what the number is. We actually care more about like orders of magnitude in some places. So I'm just going to give you a number for our purposes. Based on some of the things that come out of Landsteiner, we can say that we think that perhaps a human can recognize 10 to the 16th different kinds of molecules. Basically, you get to the point of thinking about how many combos of different like carbon atoms there are, and you can get them all with that 10 to the 16th. So this shows us just how amazing the adaptive immune system is and just how specific it is. When we think about this, then what we can realize is that if you have this whole big clunky protein, and this is the only difference chemically, the immune system is probably not viewing the whole protein together as a whole and noticing that one little piece. It's probably only binding to or noticing a section of the molecule. And that is true. And so we don't see immune responses to an entire molecule. We actually see immune responses to portions of molecules. And we refer to those portions as epitopes. So the part of the antigen that's actually making contact with the antibody, or that later will be making contact with the T cell, is known as the epitope. Here you can see we have one antigen, and it has two epitopes. It's got a red epitope that's binding to the red antibody and a blue epitope that's binding to the blue antibody. And you can see that these are sort of different types of, different portions of this molecule. Also really important, and then I'll get to your question. One other super key thing, I don't have a specific slide for it, but it is actually like so important. When we are thinking about antibodies, and this will be different when we think about T cells, so that's why I'm saying, when we think about antibodies, the antigen can be a protein, lipid, nucleic acid, or carbohydrate. It can be anything. T cells, it can't be anything. Antibodies can be anything. So a lot of the examples I'm going to show you here are proteins as the antigen. It doesn't have to be a protein. It can be a lipid, it can be a nucleic acid, it can be a carbohydrate. It's just sometimes trickier to draw those in textbook form, but in fact, all of them are possible, and it does become quite important. But Paige had a question.
So the epitope that it's binding to,
Mm-hmm.
does the antibody just know that like that's a certain part of the antigen that could be different? Like you just said it's very specific.
No, so in fact, basically you make an antibody against like every 3D structure that's possible out of things. And so I told in college, I told my roommate, who is not a science person, that immunology was really preschool because it's just about whether or not shapes match. And so it's really, the idea is that you basically make every possible antibody with every possible shape. And eventually it will find its antigen. Yeah, Jazz.
How does it figure out which configuration is best and where to locate the epitope on the antigen?
So remember, an antibody is a protein. So you guys have been very much anthropomorphizing in your question about figuring out and things like that, because it's a protein. It doesn't have a brain to figure anything out. It's really about, binding kinetics. And is the correct thing to bind accessible and present and accessible, and are you going to get the appropriate binding kinetics? So it's really all a biochemistry thing of is there access, is it present or not? But sort of relevant to that in terms of thinking about the biochemistry is that when we think about antibodies, and again, this is a thing that's key for antibodies and is different than what we see in T cells. So note that this will get different later on. Antibodies actually do care, as Jazz kind of points out, about the conformation of their epitope. So sometimes if we have, say, a protein that is folded up, so here it's folded up into a nice little S, sometimes the antibody might bind to some amino acids that are right next to each other in a line in the primary sequence. And so what would happen if you took this protein and you unfolded it, you denatured it? Would the antibody still work? Would the antibody still bind in this case? Yeah, it would. Those amino acids are still next to each other. You could stretch this out into a line instead of an S. It would still work. Everything would still be fine. This antibody doesn't care. So, but there also are situations where we might have an antigen that's like this, where it's these amino acids that are only brought next to each other when the protein is properly folded. And so you can see that. What would happen if we were to denature this protein? Would the antibody still work? No, it wouldn't. And you can also see this here, that sometimes we have an epitope that is present in the folded protein and is present in the denatured protein. Sometimes we have a situation where our epitope is only present when the thing is folded, and if you denature it, it's no longer present. Sometimes we even have a situation where the epitope is only present when you unfold. And so the conformation is going to matter a lot when we think about antibodies and antigens. The epitopes are often referred to sometimes as conformational epitopes or linear epitopes. So the conformation thing does matter a lot. If you, finish this class and say, oh my gosh, I do not like immunology one bit, and I never want to think about it ever again, and you go into some other biomedical science, this information still will be incredibly important to you, because if you ever do any experiment that involves an antibody, like a Western blot or an ELISA, or anything that involves, or if you use antibodies as medicines, they're actually the hottest kind of treatments, this is going to be one of the first things you should look at when your Western blot doesn't work. Is it because my antigen is unfolding and it's supposed to be folded, or is there an issue with this folding issue? Like, if I treat my sample and unfold it, did I just kill my reaction? Like, one of the first things so if you do experiments like that, that's the kind of thing that you should maybe know. Am I using an antibody that recognizes a folded epitope or an unfolded epitope? These kind of things become really important, even if you don't think about immunology again. One other thing that's sort of implied in everything that I've been telling you is that antigens can contain multiple epitopes. So a lot of times I like to think about the epitope as the specific target on the antigen, and each antigen might have multiple targets. So here you can see an antigen, it has 4 unique epitopes. Here and they're all different. Here you can see an antigen that has many copies and repetitive copies of the same epitope over and over again. Both totally allowed. So antigens can contain multiple epitopes. And then if you zoom out to an actual microbe, microbes have multiple antigens. And so microbes have multiple antigens, and then each antigen has multiple epitopes. So I've talked about the fact that you have an Ebola cell making Ebola antibodies right now. You actually have Ebola antibodies to every epitope on every antigen of Ebola right now. So it's not just one, it's actually a whole bunch. And I will show you this in terms of a couple of examples. So on the left, you see a cartoon version of HIV. And you can see all sorts of different things labeled. There are all sorts of different HIV proteins, and there's some lipids and nucleic acids and stuff like that. If we zoom in on this yellow protein, GP120, so that's one antigen amongst all these antigens on this virus. This virus is actually pretty small. Here's that one antigen, and you can see all of these arrows pointing to all of the different known epitopes on that one antigen. And so there are actually antibodies that can bind to all of those places on this one antigen. So we've got multiple epitopes on any particular antigen. Similarly, with SARS-CoV-2, you can see SARS-CoV-2, the causative agent of COVID, on the left, and you can see it's many different antigens. There's this yellow one that's called spike. And this was the first paper that tried to look at what epitopes were in spike. And you can see they can identify, some epitopes as a part of spike. We now know that there are about 20 antibody epitopes on spike. And so this was actually a paper and our discussion of the paper on the podcast about the fact that there are about 20 epitopes in spike. And so when you are getting COVID, so if you get COVID and you are infected with SARS-CoV-2, your body sees spike and it sees envelope and it sees this green thing. It sees all these different antigens. And you make a response to every antigen and to every epitope in every one of those antigens. If you get a vaccine, we're just giving you spike. And you're going to respond to all the epitopes in spike, and that's good enough. That's enough targets. This also becomes something where people start to debate about what's up with variants, because people are like, oh my God, if there's a new variant, spike changed, we changed the target. Usually it might change 1 of 20. So even if it changed a target, one target, the other 19 are still there. And so some people are like, look at the terrible immune change. And it's like, yes, you can demonstrate the change of the one target. But am I very worried about my vaccine not working anymore? No, I still got the other 19 targets. And this is, again, because we can think about, are we talking about epitopes or are we talking about the bigger antigens that contain those epitopes? So one other thing that I have to tell you about antibodies in general, big picture wise, is that we've been talking about these antibodies. We've been, in some cases, personifying these antibodies a whole lot. And I told you about, this great humoral versus cellular immune response controversy. But what you should know is that antibodies actually are made by a particular type of cell. Specifically, antibodies are made by a B cell, which is a type of lymphocyte. It's a type of adaptive immune cell. The best B cells who make the best antibodies are called plasma cells. And so you can see activated B cells called plasma cells secrete antibodies. So here's our B cell. It's going to interact with the microbe, and we're then going to get the production of a lot of antibodies that can deal with this microbe. It's important to mention this because really, in answering Paige or Jazz's question about things like how does it know, it gets tricky of how does the B cell know which antibody to make, or how does the antibody know what to do? And so know that really when we think about what antibodies are present, some of that is related to what the B cell did and what the B cell made. And so while, I'm talking about antibodies right now, I'm technically talking about a thing a B cell made. And we're going to basically spend some time talking about B cells, these lymphocytes we're going to talk about antibodies and sort of the details of antibodies. And then we're going to step back to the B cell a few classes later. So know that I might mess up and say something about the B cell, but the B cell is just the thing that makes the antibody. It's the cell that does the making of the antibody. Similarly, in case I mess up, B cells have a critical receptor on their surface. Immunologists are very creative. The name of that critical receptor on the surface of a B cell is called the B cell receptor, also known as the BCR. The BCR is basically the same as the antibody that B cell makes. So the cell is going to make a BCR and it's going to make an antibody, and they're going to be almost exactly the same protein. They're basically the same protein with a little bit of a modification. And so if when I'm talking about particularly what the B cell is doing and production of stuff by the B cell, I might say BCR, I might say antibody, I might mess up in some way. The reason is because they're the same thing. And the way that one gets made, the way that they're made is actually the same. And so if I happen to say, and then this happens with the BCR production, can mean the antibody, and it's all the same. And so you can imagine at this point, the BCR has its antibody, its receptor. And that's what I want you to get from this. And there is one key critical feature about adaptive immunity that you can see here. With our adaptive immune cells, like B cells, they make homogeneous antibodies or receptors. Another way of saying that is each B cell makes one receptor. So if you have 10 to the 16 different antibodies, you need 10 to the 16 different B cells. Each B cell makes one antibody and one antibody type. It might make multiple copies of it, but it makes one type. So remember how I said that there's all those Ebola antibodies you have? Each one of those Ebola antibodies comes from its own B cell. And all of your flu antibodies each comes from their own B cell. So each B cell might have multiple copies of identical B cell receptors. And that same type of thing is going to become also its antibody. And so basically the idea is you've got one B cell with each different receptor. And if one of those meets its antigen, it makes a clone army and clonally expands. And so what I want you to see here is how this is different than what we saw with PRRs. With PRRs, each macrophage has many PRRs. Each macrophage could respond to any microbe it saw. Each B cell or each antibody is unique to one epitope on one antigen. Yep, David.
So if the adaptive immune system has this secondary enhanced response, as we see in the rabbits, then how does each individual B cell response get enhanced to that antigen?
We'll get there. I cannot, honestly, the number of things I would have to explain to fully answer that question is not possible in 1 minute. But the point is that each B cell is making these homogeneous antibodies and receptors. So the one last thing I want to mention is this concept of polyclonal and monoclonal antibodies. If right now, or if you were the rabbit or whatever, you were infected with a particular microbe, you would have, or given a particular antigen like spike. Here you get that antigen, it might have multiple epitopes like blue, gray, and red epitopes. And so you'd make kind of a mixture of antibodies because you'd have multiple B cells participating in the response. And they'd all make their antibody contribution. So you get some blue antibodies and some red antibodies and some black antibodies. Yay for you. Many B cells, many clones of B cells, polyclonal, participated in this response. In the lab, we now know how to make purified versions of antibodies that only respond to one epitope on one antigen. In reality, you always are making a polyclonal response, but sometimes in the lab, we now make monoclonal antibodies. So you might have heard of monoclonal antibody therapy. And the point of a monoclonal antibody is that you have a population of antibodies that are purified. They all came from one B cell. They all respond to one epitope, and it's only something that happens based on work in a lab. It's not something that is made like naturally in an organism. But that's what monoclonal means relative to things we've been talking about before. And so we will continue talking about antibody structure as well as the function of antibodies on Wednesday. Remember that your homework is due by 5.